10 videos later, you would think that Mendel would be done with his research. A lifetime of 7 years worth of research. He figured out all of these different things about the monohybrid crosses, which is the things we've been doing so far. Monohybrid crosses because it's a cross between between one character. So you're only looking at one thing at a time. But Mendel had another curiosity. He was wondering if there was any sort of relationship between the traits. In other words, is it true that you can only be a yellow pea, or you can only, that is round at the same time, but if you are going to be green, you must be wrinkled? Is there some sort of relationship like that? That's what he's trying to figure out. On a more human example, is it possible to be smart and beautiful at the same time, and always? Is that the case? Is it the case that you are always good looking if you're smart, or if you're good looking, you must be smart? Is that necessarily true? Do traits travel together like that, or does one thing have nothing to do with the other? That is the question that Mendel has, and he wants us to try to answer it. Now, just like I did with the other crosses, I'm going to give you the answer first, so that as you go through it, you can look for how he found that answer. He actually found out that yes, it is completely independent from each other. There is nothing to do with each other. One trait travels the one way. You are smart because you're smart. It has nothing to do with the fact you're good looking. You know, so being like this is a very lucky situation, because you're not going to be good looking and smart all the time. So not everybody is this lucky. You know what I'm saying? I'm just kidding. Clearly I'm not good looking or together. But the other that he's trying to figure this out is by doing the same kind of thing he did with the other crosses, but trying to do it with looking at two traits at once. So he gets his data and sees if there's any relationship between looking at two traits at once. Now, he didn't have to do this experiments from scratch, because he already had the data collected, and he wrote everything down for every single flower. So he just went back to his data and did analysis on his data to figure out if there was some sort of pattern. So look what he actually finds out. He finds out there is no relationship. Now, he was actually wrong about that. We have 23 chromosome pairs. Now, the reason why there is independent assortment is because of meiosis. And we're going to talk about that in a second. But meiosis also proves that some genes are going to be in the same chromosome, so they are going to travel together. So he wasn't exactly right, and we'll talk about that later. But let's pretend that you are Mendel and you look at your data, and for the most part, he was right. There's exceptions to this rule, but for the most part, he was right. So how did he do this? He got a plant that was completely dominant for both traits. So that means a plant that was a yellow pea that was round. So that means it's dominant for both traits, because roundness is dominant and yellowness is dominant for the color of the pea. And so then he gets a pea that is green and wrinkled, which is the recessive for both traits. So what he's doing is a pea cross. Now, I'm not going to do that cross because it's really simple. So I'm going to keep that away. And if you want to do that cross later, after you see how to do a dihybrid cross, you can do it for practice. But what he does next is what's interesting for me. Because what's interesting is that he gets the same kind of result that he got when he did the monohybrid crosses. This time he's doing two traits at the same time, so it's not a monohybrid, it's a dihybrid cross. So it's a multi-character cross, when he's following two characters at once. But this time, same thing, he gets something that's hybrid for both traits. So this animal is going to be hybrid for color and for shape, which means he's going to be dominant for both, right? Because dominance dominates when the presence of a dominant gene causes you to look dominant. So this is going to be all the seeds were yellow and wrinkled when he did that cross. Then he did what he did last time again. He got that and crossed with itself and with other members of the same generation. Now what is interesting. What he actually got is a 9:3 to 3:1 ratio of looks. What that means is that on this F2 generation, he got 9 that looked dominant for both traits, 3 that look dominant for one and recessive for the other, 3 that look recessive for one and dominant for the other, and 1 that look recessive for both. That means you can have a green wrinkle thing, but you can also have a green round one. You can have a round yellow seed, but you can also have a yellow wrinkled pea. But that seems to happen in some sort of numerical pattern. 9331. So now he wants to understand why this is happening. And so he does a series of experiments to try to understand why this is happening. Now, if with any other cross, you always have to set up the top, like we talked about. You do the character, the alleles, the genotypes, the phenotypes. So let's look at that. We're going to be working on this series on the seed color, which has alleles big Y, which means the dominant yellow, and little y, which means the recessive green. So when you get big Y and big Y, you make the dominant homozygous dominant look, which is yellow, or big Y little y, which is the hybrid or heterozygous, is also going to look dominant for yellow. And then you have the little y, which is the only way to look recessive, green. Now, the same thing we have to do for the other trait. In this case, we're going to look at the same time at seed shape. And for seed shape, big R means dominant round, and little r means wrinkled, which is the recessive. Now, I would like to point out that obviously round was discovered first, because if round was discovered second, W would be the letter that we use for the trait. Remember, capital means dominance, but the letter that's chosen means whatever was discovered first. And so that's how we know that round was discovered first. In this case, the dominant was discovered first. But it is possible for you to have a letter that does not necessarily match what's the dominant. So you could have big W to mean round if wrinkled had been discovered first. So make sure you know that. Now, anyways, that big R is going to mean the dominant round. Big R little y is also going to be the dominant round. And little y will be the recessive wrinkled. Now, keep this in mind as we're going to go through the traits, through the cross. Now, what the cross we're going to be doing is that F1 cross, the cross between those two hybrids. In that cross, basically what he does is he gets something that is a big R little y versus big R little y. That's what we're talking about there, that middle cross there, between two things that look the same, a self-pollination of the F1 generation. So, obviously, he's going to be looked dominant for both traits. Like we see in the picture, there's going to be round and yellow versus round and yellow. So, now, what can you make? Now, think about this. What kind of gametes can you make? There's two traits. So each gamete is you have to look at both traits. So you're going to make 2 letters per gamete. It's not like the other one. Remember, everything is doubled here. It's a dihybrid cross. So for one gamete, this big R can combine with the big Y. For the other gamete, the big R can combine with little y. And this is random, the way it combines. Remember, independent of assortment and separation of homologs doing anaphase 1. We're going to talk about more about that later. But that means, I'm sorry about that. My dogs felt like they had to contribute to the video. And so, but anyways, you're going to get, what that means is that you can get a random distribution of them. So you can get the big R with the big Y or the big R with the little y. Likewise, you can get the little y with the big Y or the little r with the little y. So let's see what I'm talking about here. Basically, if you combine the big R with the big Y, you make this gamete. But you can also make this by combining big R with Y. Also make little r with the big Y and the little r with the little y. So there's 4 types of gametes you can make from that. Just like math when you do distribution, right? So that's how you do that. And remember to always do this step when you do a dihybrid cross. Otherwise you screw up the way you set up the Punnett square and it doesn't really work well. So if you're actually going to do a dihybrid Punnett square, even though I'm going to teach you later a way not to do it, you should always set up the possible gametes ahead of time. In other words, you set it up wrong. But the second thing is going to be the same thing because it's the same organism. So you're going to basically have the same exact gametes on the other side. So that's what you got. So I hope you understand why it's like that. Because remember, it's 2 traits you're following. So instead of just doing one gamete, you're going to be, it's still one gamete, but it has 2 traits you're following in each one of them. So let's look at the actual cross. We're going to be crossing 2 hybrids for both traits, which look dominant for both traits. And they're going to have those possible gametes that we talked about. Since they're the same, they're going to have the same possible gametes. Now, just like the other Punnett square, you're going to put one gamete on next to each box. Now, notice that we have 4 rows and 4 columns. Before, when we only had 2 types of gametes, we had, like for big A and then little a, it was only, when you separate that, you can only make 2 types of gametes. One with a big A, one with a little a. And so, because you're only looking at one trait, you can only make 2 types of gametes in that sense. You're going to make a 2x2 Punnett square. But since in this case, you have 4 different kinds of gametes, you need 4 rows and 4 columns on your Punnett square, and that's where it gets complicated. But anyways, you're going to put one gamete next to each box, just like you did before. So you put the big R, big Y there, the big R, little y there, the little y, the big little r, big Y there, and then that one here. And you do the same thing on the top with the other gametes from the other parents. Now, you do the same thing you would do for the other Punnett square. You combine the letters which you see here. So, for example, on the first box, you would have to put a big R, another big R, another big Y, and another big Y. And you always put the characters together. All the R's together, all the Y's together. So, just to accelerate the video a little bit, I'm going to do a bunch all at once. So you see, boom, you have all of them there. Now look at that. Let's look at the first row to see how I did that. Big R combining with big R. So you got this big R coming here, and you got this big R coming here, and then you got this big Y going there, and that little Y coming here. So that's, you see how they're combining on the thing. And the same kind of thing will happen on the next one, where the big R will combine here, right? And you're also going to get the big Y is going to combine here. And then you're going to have a little R combining there and a big Y combining there, and then you get that thing. So you basically do the same thing you would do for the other one, except you're doing for two traits at once and setting this up like that. Okay? Now, why do I put all of these specific 9 things at once? Because look at the pattern here. Look at the first letter of the thing. The first set. All of the first ones are dominant. Dominant, dominant. Now, this is a hybrid, but it's also going to look dominant. So these are all going to look round because it's the dominant look. You see? What about the second trait? It's all yellow. So that means all of these things are dominant for both traits. And so all of these will be round and yellow. And that's why you get 9 of them, like I said on the previous slide. Now, what about this box here? You're going to get a big R with a little y, right? And so these 3 boxes, what's happening here? You get a big R, so that's a dominant for the first trait, dominant for the first trait, dominant for the first trait. But the second traits are all recessive, which means it's going to be round and green. What about the next 3 boxes over here? The next 3 boxes will be little r with big Y. So you're going to get something that's recessive. These are all recessive for the first trait, but dominant for the second trait. So you're going to get what? Something that is wrinkled, but yellow. And then only the last one. Do you ever combine all the recessives into one to make the recessive look that is green and wrinkled? And so that is why you're going to get 9 that look round and yellow, 3 that look round and green, 3 that look wrinkled and yellow, and 1 that looks wrinkled and green. And basically what you see here is a 9:3 to 3:1 phenotypic ratio. And this will always happen anytime you see a dihybrid, which is hybrid for both traits, crossing with another hybrid just like it. You're always going to get a 9:3 to 3:1 ratio. What that means is that it's always going to be something that's dominant for both traits 9 times out of 16. So 9 out of the 16 boxes were dominant for both traits. Then you're going to get something that was dominant for the first trait, but recessive for the second trait, 3 out of the 16 boxes. And then you're going to get something that was recessive for the first trait, but dominant for the second trait, or 3 out of 16 boxes, and then 1 that was recessive for both traits, or 1 out of 16 boxes. In other words, 9 will be dominant for both traits, 6 out of the 16 will be dominant for one, but recessive for the other, 3 each way around, and 1 will be recessive for both traits. And since this is what happens, because there is something that's called independent assortment. The traits don't really talk to each other. And I talked about it, you see why that's the case. It's not any other thing other than that, because you see that it's completely possible to get the traits with different combinations. You don't necessarily must be green if you're wrinkled, or must be yellow even though there seems to be a predilection for the dominants to be together than the other way around. Alright, so this is a dihybrid cross, and on the next video we're going to practice 2 more dihybrid crosses so that you can get a picture of how to do this, and they will be simpler than this one. And this is the one you have to be able to recognize out of the top of your head. 9331, that means 2 hybrids for 2 traits matter with each other. Alright, see you on the next video.